altering a mosquito's gut genes to make them spread anti-malarial genes to the next generation of their species shows promise for curbing malaria. This is the finding of a preliminary study by researchers from Imperial College London and published in the journal eLife. Mosquitoes are increasingly becoming resistant to pesticides, and the parasite that causes malaria is also becoming increasingly resistant to anti-malarial drugs. This has created an urgent need for new ways to fight the disease, which in the year 2019 caused an estimated 229 million cases and 409,000 deaths, most of which were young children in sub-Saharan Africa. Gene drives are currently being tested in labs as a new approach. They work by creating genetically modified mosquitoes that, when released into the environment, would spread genes that either reduce mosquito populations in a particular geographical area or make the insects less likely to spread the malaria parasite. Malaria is caused by the parasite Plasmodium, which certain species of mosquito carry in their guts. The team genetically modified Anopheles gampia mosquitoes so that after they have taken a blood meal, they express small antimicrobial molecules that target and impair the development of the Plasmodium parasite. They initially inserted the gene along with a fluorescent marker to help them track it in 3 different spots in the DNA, and then later removed the marker, leaving only a minor genetic modification behind. Next, the team bred the mosquitoes to see if they were able to successfully reproduce and remain healthy. They also tested how well the malaria parasite developed in the mosquitoes' guts. Their experiments provide preliminary evidence that this approach to genetic modifications could create successful gene drives. The team's modification essentially hijacks a functional gene in the mosquito to produce the antimalarial molecules. While this modification can be passed on to the mosquito's offspring, at present it would only be passed on to some offspring. The researchers say this means their approach simplifies the testing of the antimalarial effect using these mosquitoes in the field, as the genetic change would only be passed on to a small proportion of mosquitoes in a local population in any preliminary tests. A full gene drive would ensure that the modification would be inherited by all offspring. This would ultimately require the addition of a specific component called Cas9 once safety and efficacy had been established. These genetic modifications are passive and could be tested in the field and undergo a stringent regulatory process to ensure they are safe and effective in blocking the parasite without raising concerns of accidental spread in the environment.